Hello, everyone. I hope you are doing great. First of all, I would like to say thanks to the organizing committee of this conference who gave me the chance to share some of my research work with you. My name is Tahir Qamar. I finished my PhD Bioinformatics last year and currently working as a postdoctoral fellow in Guangxi University, China. Today, I will briefly introduce you with a pipeline I developed during my PhD research. It's ppsPCP. The purpose to develop this pipeline was to help the eukaryotic research community in developing the pan-genomes. I used couple of programming languages during the development of this pipeline, and this pipeline give you the fully annotated pan-genome that can be directly used for various research purposes. For example, the gene level analysis in molecular breeding or biotechnology work. It will also help you in your academic publications if you are working with the whole genome sequencing or pan-genome related work. Why we need pan-genomes? There are a couple of studies published that highlights the importance of pan-genomes. I'm giving you the example of a paper published in Nature Communication in 2015, where the authors compare the tropical and temperate maize line genomes, and they found that 50% of their genome is structurally different from each other. So just imagine if you're using a single reference genome to study the diversity or genomic content of a species or of individual, all individuals of a species. Same like this, you will lose the half of the information or most of the information during the process. So that's why we need the pan genome, because the single reference genome cannot capture the entire genomic content or the diversity present among the members of a species. A couple of pangenomic solutions were available for the eukaryotic pangenomes, but none of them was able to give a fully annotated pangenome, and they have their other limitations. So keeping in mind their limitations, we develop a new protocol. And in our protocol, if I say in simple words, it consists of the four basic steps. You need to select a reference genome, already established reference genome. And then you can compare your query genomes, multiple query genomes with that reference genome. Our pipeline or the method, the developed method, will extract the PAVs from the query genomes and merge them with the reference genome to develop a new pan genome. This is the simplest explanation, and we submit our protocol patent to the China Intellectual Property Right Administration, and it was accepted. And then we transform our protocol into a pipeline to give the benefit to the maximum people. And our work has been published last year in Oxford Bioinformatics. You can read more details online, and you just need to supply the basic information in the form of the FASTA file or the GFF3 annotation file to this pipeline, and it will develop a fully annotated pangenome for you. um, our software, or you say the pipeline, is freely available on its web page and also on the GitHub page. The links are here. You can download free of cost. And this is the front face of PPS PCP pipeline. It's a single command use pipeline. Anyone can use that. You don't need a prior bioinformatics skills or computational biology skills to understand or run this pipeline. Anyone can read the manual or the tutorial available in the web page. We describe all the steps, and we enlisted all the instructions. You can use this pipeline very easily. And there are also a couple of thresholds, cutoffs, parameters are available. You can use those according to your requirements or research purpose. This is the basic algorithm, and I will not go into the detail of the every step due to the time limitation. And but systematically it gives you a complete annotated pangenome. uh, we developed the pan genome and we tested with multiple plants genomes. As our main focus was on the plants, so we tested first with the plants. In a paper published recently in Nature Biotechnology, where the authors develop a pan-genome for 18 Arabidopsis ecotypes. We use the same data to develop the pan-genome with our pipeline, and our results were better. Our method develop a pan-genome with more than 1600 genes, and also in a very less time. It takes only 10 CPU hours to develop the gene, the pan-genome. And furthermore, we also tested our pipeline with the rice data. We considered the Nipponberry, Shuhui, and Minghui, three famous rice species, and developed the pan genome using our software. And our pan genome had the 43,000 genes. And then we compare our developed pan genome with reported reported pan genome of rice. The paper published in 2018 in Nature Genetics, and the authors developed a rice pan genome from more than 3,000 rice sections. So our three species pan genome, we compare with their pan genome, and in a result we see that more than 41,000 genes were fully mapped to our pan genome. So that shows that high accuracy of our developed pipeline, and it takes only 25 CPU hours. So this shows the high accuracy and the high utility of our pipeline. So this was the basic introduction I gave you, and one of the purpose to give this talk in this Applied Zoological Society of Pakistan was this pipeline is equally useful for any kind of data. You can use the prokaryotic data, you can use the other eukaryotic data. For example, you can use the human data, you can use the animal data to develop the pan genome. And so you can use as far as the file formatting formats are same, you can use any kind of data. So you can use your animal data, and you can use the develop the animal pan genome. Here is the example of some authors from India. They use our pipeline to develop the pan genome for their bacterial target, and they successfully published this paper in Frontiers in Microbiology. So this was all about my talk, and this is my research group, and our research was funded by National Science Foundation, National Natural Science Foundation of China, China Scholarship Council, Huazhong Agriculture University, and Guangxi University. I thank all of them, and I also thank you for your attention and listening. If you have any question, you can write me email, or you can raise your hand here. Thank you so much.